Evening, everybody. We'll just wait for the numbers to go up so people can join, and then we'll start tonight's webinar. Okay, it's 7 o'clock, and we'll make a start. Thank you for joining us tonight on this NSA SPV Schmallenberg virus webinar. I'm Kevin Harrison. I'll be your chair for this evening. I'm a sheep farmer down near Bath, and I'm also chair of the NSA's Policy and Technical Committee. We have a couple of great speakers lined up for you tonight. We have Dr. Christopher Sanders. He's an Institute Fellow at Pirbright. Covers entomology, epidemiology, and virology. His research focuses on the physiological and the behavioral attributes that enable an insect species to transmit a pathogen, or in other words, a vector capacity, since 2006. His work has explored the behavior of biting midges, vectors of viruses between ruminants. We also have Rudolph Reichel, who has been the Small Ruminant Expert Group veterinary lead since January 2023, having taken over from Amanda Carson, and he leads the DEFRA-funded Small Ruminant Disease Scanning Surveillance Project in AFA and is a committee member of the Sheep Veterinary Society. Rudolph also represents AFA regarding small ruminants on the Ruminant Health and Welfare Group. So we'll go straight into the presentations, which I believe, Rudolph, you're going to go first for us. Whilst you're getting that up, we'll do the questions at the end. So if you've got any questions to ask, please feel free to type them. If you could put them in the Q&A rather than the chat, that would be great, because then we can just sort of manage them a bit better rather than in the chat. Thank you, Rudolph. Over to you.
Okay. Is my screen shared okay, Kevin?
Yes, all up.
Okay. Right. So thank you very much for the opportunity, and good evening to everyone that's dialed in. So just a brief introduction about Schmallenberg virus. So the initial cases of the recent outbreak, we started to get reports in November and December 2023 in England, and there's milk drop in dairy cattle, fertility issues in sheep flocks, high barren rates, poor scanning results, and then there was also positive serology results on bulk milk testing from dairy cows, and then also on sheep due to be tested for export, for germplasm or semen export. So then, end of December, start of January, we started to see stillborn and deformed lambs being born, and they were confirmed on PCR, and the first cases were seen in the counties of Devon and Dorset. So a bit about the background. It's still a new virus, first described in 2011, compared to some of the other viruses that's been around for decades or centuries. So it's in the Orthobunia virus group, and there's over 170 of them worldwide. We can get similar viruses found in Australia, Africa, and Asia. So this one affects predominantly sheep. Cattle and goats are also affected, but in lower numbers. And the early infection can cause diarrhea, high temperature, milk drop in some adult cows, and then they recover within a few days. So we don't tend to see that, excuse me, in sheep or goats. The striking thing we see is the deformed newborns and abortions, and then barren ewes, and that is then also the main issue and concern to us. And the virus is transmitted by midges from the Coides group, so similar to blue tongue. So the original outbreak in 2011, there was an undiagnosed syndrome with diarrhea, high temperatures, and milk drop in adult dairy cows in the Netherlands and Germany. And then it was confirmed there. And then in January 2012, the first deformed lambs were born in Norfolk, Suffolk, and East Sussex. So we had cases here as well. So the map here on the right just shows the distribution from the start there, and then by 2019, it included a much wider area. So it's distributed up to Eastern Europe, Turkey, Greece, and Russia. I'll just enlarge the map a bit so one can see it clearly. And it colors just shows where it started in 2011 and then gradually spread further and further east and further north, and covering a vast geographic area. So this map here is what the clever people do with the wind maps and risk assessments. So if you have infected areas here, and it is in the mid-season, and you've got wind blowing in the right direction across the channel, then they can predict that some of these infected midges would be blown over. And as you can see, the high-risk counties here are then on the east and the south coast, where they're likely to get hit by infected midges that can then transmit it to susceptible animals. So that's how it started, but obviously now it's become endemic in many areas. So it doesn't need to be transferred again from Europe. We've got it here now in animals, and it pops up every now and then. So when we had the original outbreak in 2012, the first cases, again, as pointed out by that risk map we saw here in the eastern counties, and then gradually spread further inwards and northwards as well. Although some of that spread is sometimes due to the time of when animals are tupped. So further north you go, they tup later, so you're going to see the signs later as well. So some of that spread is a bit artificial. So a bit about the transmission. So we know biting midges are the route of transmission. They're particularly attracted to cattle, and we're not aware of other biting insects that can transmit it. There's no direct animal-to-animal transmission apart from the dam to the offspring in the uterus, if she's infected at a certain susceptible stage. We don't know about persistent infection. It's not been confirmed. So it's unlike diseases. We see reported disease or BVD, where animals can be persistently infected for lifelong periods. We don't know about the embryo in infected animals, if that can potentially carry the virus and be transmitted over. Semen, there has been some positive semen detected in AI bulls, and they were detectable there for 3 months, but it hasn't been proven that you can transmit the virus that way, but that is a possibility. I'm not aware of any good studies that's been done in sheep. So I guess it's one of those areas that in the back of their mind, there's something that needs to be confirmed or proven or disproven. And then there's no evidence of oral infection, so not via milk or via ingested amniotic fluid or placenta. The main route is the midges. So the clinical disease, the main losses we see in sheep is early embryonic death. So you get these poor scanning results or high barren rates. Then what we can see more easily is the abortions or stillbirths, some without deformities, some can be mummified. But then the clear thing is these horrible deformed lambs coming out, and some with brain damage, those twisted rigid legs, twisted spines, deformed skull and jaw. Some can be normal, but then they've got this dummy appearance because they're brain damaged, or they can be weak and don't thrive as well as you expect them to do. There's also the milk drop in dairy animals during the acute infection, which can be seen in cattle, sheep, and goats. It's mainly picked up in cattle. But we do see a large variation on individual farms following natural infections, and all the factors involved are not known yet. So the timing of infection for stillbirth deformities to occur, and depending which papers you read, there's slight differences there in those dates. But roughly in sheep, it's between 1 to 2 months, and then in cattle, 2.5 to 5 months, where you've got the susceptible period. So it's important to remember that if you get a deformed calf born, then it's 4 to 6.5 months ago that the dam got infected, and sheep, it's 3 to 4 months ago. So that sometimes makes it difficult to know where the animals were exactly and what were you doing or not doing So, and what the weather was at the time, or were they housed or not. So the impact of disease in sheep flocks, we mentioned the increased repeat breeders or increased barren rates. Then the lamb losses from early absorption to abortion to stillbirths. You can see increased lamb mortality or lambs not thriving. And then you can get ewe mortality as well from dystocia. Obviously, that can increase your veterinary costs as well if you have to do cesareans. And I think the one thing that is difficult to quantify is that impact for the livestock keeper when they have to deal with that and see that, because it's one of those conditions. It's an awful vision, and there's nothing you can really do. There's no medication you can do at that point to correct it. And then the next lamb, you're on tenterhooks to know if that is going to be the same or if it's going to be normal. So some of the deformities we see in these lambs is a flattened skull because the brain isn't fully developed. You get that lower jaw or the parrot mouth appearance. And like we said, with the brain, it's sometimes like a balloon, fluid-filled balloon. So if you cut into it, it just flattens, falls flat. And then if you know the anatomy, but you should have what we call the small brain there, or the cerebellum, and it's very small. It should be filling that space, and same here, and it's just almost absent. And then your, call it your main electric cable to take the signals, so your telephone cable taking the signals from the brain to the body, is very, very thin, very underdeveloped there, very small. So the signals doesn't go to the rest of the body, and then the fetus stops moving, and that's when it becomes twisted and rigid. And then we see with this twisted spine and these awkward shaped limbs that can cause dystocia, and that just looks awful, especially if they're born live. The other thing we had this past season is that we had Schmallenberg virus and blue tongue virus, and in some places it was in the same areas at the same time. So the problem there is that both intrauterine infections can cause similar lesions in the brain of calves and lambs, and you really have to test to differentiate the two. You can't just look at them and know for sure which disease was involved. So we encouraged investigations and submissions to rule out blue tongue virus and to confirm Schmallenberg virus, and also to make sure that we're not dealing with a new virus or a different virus. And because blue tongue virus 3 is a new virus, we were not quite sure if it could cause this twisted limbs and twisted spinal columns that we see with Schmallenberg virus. And IPHA also offered free Schmallenberg virus testing to facilitate this. So immunity, and there's just a paper where I got those figures from. They mentioned that Schmallenberg infection, so this is a natural infection, will induce a solid immunity in cattle and sheep. And in sheep, it should last for 4 years, and cattle 6 years. So once they've had it, and if a good number in your flock has been exposed, then in theory, you should be safe for a number of years. So the vaccines work, but I have to say if available, because currently we do not have a licensed vaccine available here, unfortunately. So just about some of the cases we detected in the past season. So we're going to look at the period from 1st November 23 till January this year. And keep in mind, when you look at the map, the database only covers England, Scotland, and Wales. So it doesn't cover Ireland. And so we had a total of 366 confirmed submissions, 102 calves, 2 goat kids, and the remainder lambs. And just to say, when we say submissions, it could be sometimes one submission is 2 or 3 affected lambs. So the number is higher. Then the other thing on the map, the darker shades indicate that there's a higher number of submissions in relation to the number of sheep present in that county. And then the other thing to remember is that submission of these cases and the reporting is voluntary. It's not a notifiable disease, so we wouldn't know of all the cases that were there. So there's probably going to be quite a significant under-presentation of the real impact of this disease that we had this season. So but that's the map there. So this shows the counties where we had confirmed cases. And so like there on the Isle of Wight is quite dark. So we had quite a lot of cases in relation to the amount of animals that were there. And again, some of the darker shaded areas had more cases than others. And some of the lighter ones probably only had 1 or 2 cases like that up there in the north. But we can see it's sort of over Wales, over to the west side. And with some of the counties here didn't have any cases in this period. So just a bit of a look back to when it started, and we get this interesting pattern. So when we started in 2012, we had this peak here, obviously, and then a bit in the next year, but then it sort of disappears. You see very few cases or almost nothing. And sorry, the green line here is sheep and the blue line cattle. So then in 2017, we had another peak, as was predicted. And then again, it disappeared. And then we had a bit of a blip in 22. So we sort of thought that was our peak and that's going to be okay for the next 4 or 5 years. But then this happened. Quite a lot of cases and quite high numbers. And our thoughts on that is, if we look at these maps, so this is our current outbreaks or the past season, and this was the previous peak. And it looks like it was only a few cases and they were more focused on the east. So if you look at Wales there or Midlands and North England, they had no cases. So you can imagine those animals there hasn't had any cases for quite a few years. So almost all the animals there would be susceptible. And I think it probably just spread from there further on. And that's why we saw so many cases this year. And it probably coincided with just ideal conditions for midges and for the virus to spread. And we had a long, warm autumn, so it carried on for a long time, unfortunately. So I'll stop there, and then we can pass over to Chris. If I can just find the right button. There we go.
Thanks, Rudolph. I'll share my screen now. So that's... Are we good? Can you see that? Yeah. Brilliant. Okay, so Rudolph's already introduced culicoides and as vectors of Schmallenberg virus. Oh, come on. There we go. So these are culicoides biting midges. And so one of the, there's a few characteristics that they all share that are important for their ability to spread viruses such as Schmallenberg virus. Culicoides are amongst the smallest blood-feeding insects. They really are tiny, 2 to 4 millimeters long, much smaller than your average mosquito. They have a near worldwide distribution. There's about 1,500 species worldwide, about 50 in the UK. Typically, culicoides can be hugely abundant. And by that, I mean we can collect tens of thousands, if not hundreds of thousands, of insects per night. The international record for a trap catch of culicoides comes from South Africa, where they caught more than 2 million individual insects in a single night. I fortunately did not have to count them. So there's huge numbers of them, and there's quite a few different species, but only a few are actually vectors of viruses such as blue-tongue virus. And helpfully for us as entomologists, culicoides have these patterned wings, and we can tell vector species such as this one from non-vector species. So this one is Culicoides obsoletus, and it bites sheep and cattle quite readily. But this is Culicoides circumscriptus, and that bites birds, so it wouldn't be a vector of Schmallenberg or indeed blue-tongue. Culicoides, unlike mosquitoes, don't breed in free water. So a lot of invasive mosquitoes and native mosquitoes breed in ditches and ponds and anything that water can collect in, like old troughs and things like that. Culicoides don't do that. They breed in damp organic matter. So they still need moisture, but it has to be in a substrate. So things like soil, leaf litter, leaf mold, and animal dung. And different species live in different habitats. And the females, and only the females, require a blood meal to produce their eggs. And this biting habit leads to their role as vectors of viruses. So their eggs are laid in these moist organic substrates, which are found throughout farmland. So farms present everything that a Chilocoides midge needs. So the eggs develop into larvae, which are little worm-like larvae, look a bit like nematode worms. They pupate and then emerge into adults in the spring. So they overwinter as larvae, so they're in the ground essentially as larvae through the winter, emerge in the spring and summer. And then the females undergo this kind of cycle of blood feeding and egg laying. And obviously this means that if they bite an animal, they become infected and then can transmit viruses. So the transmission cycle between midges and animals, we start off with our viremic infectious ruminant host, and a midge bites that animal. And some midges, not all, but some midges will pick up that virus, and then the virus has to replicate inside the midges themselves. This is a temperature-dependent process. So the warmer it is, the faster this will happen. As I said, not all midges are actually capable of replicating this virus. There's lots of different barriers to infection of the midges. But because there's so many midges, even a small proportion can actually go on to transmit the virus. So once that incubation is complete, the virus has reached the salivary glands, and then when that insect then bites its second meal, if that's a susceptible host, it can then transmit that virus. Now, for bluetongue virus, we know that a single bite from a single midge may be sufficient to transmit the virus, and that animal will develop a full viremia. We don't know if that's true for Schmallenberg virus. One thing we do know is that Chilocoides are a little bit more efficient at picking up the virus, picking up Schmallenberg virus, than they are bluetongue. After a susceptible host is bitten, it'll go through a latent period of a couple of days before it then becomes infectious. And one of the differences between Schmallenberg virus and bluetongue virus is that the host is actually only infected for a very short period of time. And this is one of the contributory factors determining the pattern of infection we see. So our animals, once bitten, will actually only be viremic for between 2 and 5 days, so very short acute infection period. Typically, as Rudolph was saying, we don't see signs in these animals that they have been infected. We may see fever, diarrhea, and milk drop in the cattle. But it's these fecal malformations happening early on in pregnancy, as Rudolph was saying. And this short infection window determines whether we see disease or not, because it depends when these animals were infected during pregnancy, when the virus was present in that area, to whether these animals were pregnant at a susceptible period of pregnancy, to whether we actually see a clinical disease. So animals can be infected and not show any signs at all. But if some animals were pregnant at just at that wrong time, then we see the malformations and the horrible impacts that we see of Schmallenberg virus. Whereas others, we won't see any signs at all. As Rudolph was saying, infected animals gain long-lasting immunity to Schmallenberg virus. And that means we actually see cyclical outbreaks of Schmallenberg. So it erupts on a cyclical basis. This is due to decline in herd immunity. As these animals get old and replaced in the flock or herd, new animals are born, obviously, and they won't have any immunity. And so you get to a point where the infection pressure is such and the immunity is dropped so much, we get these outbreaks somewhere on a 4 to 7 year cycle. So Schmallenberg virus transmission is seasonal, and that's because it is transmitted by Chilocoides, who are themselves seasonal. So Chilocoides adults are active between April, they kind of emerge as in the spring. We get a peak of activity in April, April to May to June, and then we get a second peak in sort of September, October. Chelicoides are active at dusk and dawn, and they're crepuscular. That's why we can collect them in light traps. We have a network of light traps which tell us what's going on in the midge population in the UK. And we know from this trapping that their activity is strongly influenced by the weather. So when it's warm, when it's humid, and when it's still, these small insects are far more active. When it's too windy, they're not very active at all. When it rains heavily, they're not very active. But when you get that fairly dry, warm, yet humid weather, they can become very active. When we see higher temperatures, that affects everything to do with the midges is temperature, is related to temperature. So they'll develop quicker, they'll be more active, and they'll bite more. And so there'll actually be more midges when it's warmer. And also because the virus replication in the midge is also regulated by temperature, there's going to be more infected midges when it's warm as well. So we see this autumn peak in Chelicoides activity, and that corresponds to this increase in virus within that vector population as well, and also unfortunately with tupping and pregnancy. So therefore we get this kind of perfect storm of increased midge numbers. It's typically warm that time of year, so there could be more midges that are infected that time of year, and it's also at that peak susceptibility. Both the Chelicoides and therefore Schmallenberg infection declines in the winter. When we start getting those first frosts in the autumn and winter, we suddenly see the adult midges drop. Where Schmallenberg virus goes in the winter, we don't know. So it keeps coming back, obviously. So it's still present in a population, maybe at a low level that we cannot see, or in the midge population, and survives through the winter to then infect animals again in the spring. So what can we do about Schmallenberg virus? Essentially, what we'd aim to do with vector control is to reduce the contact between animals and midges. And the first thing to say is there is no way to protect all animals from being bitten or prevent all bites. These insects are everywhere and they're very abundant. There's no single product that we can use to stop Schmallenberg virus transmission. There's no way we can treat the animals other than through vaccination. With blue tongue virus, we know that a single bite from a single infected midge can reliably transmit the virus, and that's possibly true for Schmallenberg virus as well. If it's not a single midge, there's certainly evidence that only a few are required. So how do we break this cycle of transmission? Well, there's been a number of methods and techniques and protocols that have been suggested, including sort of removal of larval habitats like dung or mass trapping midges to remove them from the environment. Because of what I said about the larval habitats of Chelicoides, they're present in all sorts of habitats across farms. So removing one habitat like a dung heap, for example, doesn't actually impact on Chelicoides populations. So, and because it's spread across the whole farm, we can't use insecticidal spraying as you might use for mosquitoes or other insects, because you would be spraying such a wide area. So we cannot recommend that. Again, midge traps are not going to be able, no matter how big your midge trap is, they're not going to be able to collect sufficient numbers of insects to dent that population. So removal of larval habitats and midge traps have not been shown to influence Chelicoides populations at all. What about what we can do with the animal? How about repellents that kind of change the smell, the odor profile of your animal to stop midges being attracted to them in the first place? So we're aiming to repel Chelicoides to prevent that contact with the animal. So you might be familiar with using insect repellents yourself if you get to the tropics, or indeed to go to Scotland in the midge season. Slightly different species of midge, the Scottish biting midge, so the ones we're concerned with, but it's that level of biting that we're talking about. So you've experienced that where they're in your eyes, in your ears, up your nose, horrible. And if you've been there, you'll know that if you use insect repellents, you have to repeatedly apply them. And the same is true if we would use these for treating cattle and sheep. Their effect is going to be very transient and lasting perhaps only hours. And so you'd have to repeat application on a very frequent basis for these to work at all. And they've not been shown to affect Schmallenberg virus transmission in the field. The same is true of synthetic pyrethroids. So synthetic pyrethroids in pour-on formulations have been shown to kill culicoides in the laboratory, but may not prevent transmission to that treated animal because the insects can still get to the skin, they can still bite, but they might die sometime afterwards. They're a little bit more persistent than repellents, but lasting days rather than the weeks we'd need. So again, you'd have to have repeated applications of these to work, and no product has been shown to affect Schmallenberg virus transmission in the field. We also have the problem with pour-on insecticides, that the insecticide doesn't actually get to where we really need it to. So a lot of these products are designed to be poured along the back line with the aim that they would gradually seep through the fleece or fur, covering the flanks. Culicoides midges like the more easy to get to places, the slightly thinner skin around the ears, between the legs, around the legs, and the neck of these individuals. So we're not actually getting that active product to the areas that are going to be bitten by culicoides. So another technique we can use is actually sort of move your animals or shelter them in some way that again removes them from the midge biting areas. And again, the biology of midges means that there is something we can take advantage of here. If you've got grazing in exposed areas that are quite windy, a lack of tree cover, for example, those areas are going to have less culicoides for less time than areas where you have an enclosed space which is going to retain humidity and it's not going to be very windy. So take advantage of those areas, put animals out there during a susceptible period if at all possible. Stabling has been suggested to prevent culicoides reaching animals. When we talk about a stable, one person's shed is very different from another. And typically most stables and shelters are open in some way to insects. So I've got a picture there of a barn that has a whole side that's open, and midges will definitely enter there. Culicoides, some don't prefer to go into stabling, but they certainly will do, especially in the autumn. When it gets a bit windy and a bit wet outside, if you've got animals inside, they will actually come into those shelters. We can arrange our shelters to make them vector proof, but this is a really significant challenge to do. It's possible with screening using mesh with a very small aperture, so very small holes. A lot of the products that may claim to be midge netting are not, just don't have holes small enough, and these insects will still get through those holes. So very small aperture mesh, and screening all the holes that you can. Increasing ventilation. So we know if it's windy, we get less biting. If you can increase ventilation in those sheds and basically push insects out. So if you can generate an airflow through your housing of an airflow greater than 3 meters per second, which is actually quite fast, you can actually reduce biting. With stabling, though, we have to consider the welfare implications of the animals as well. So sticking animals inside a shed during very hot periods is also going to be problematic. And you've also got the increased feed costs and things like that. So it is difficult to achieve and has implications for costs and welfare. And it may also be counterproductive. If you're sort of housing all your animals in one place, and that happens to be somewhere that midges can get to, you may actually concentrate all those animals in an area with infected midge population, and they all get infected at the same time. So there's lots we have to weigh up, and it's not a simple answer. With regards to transportation, there's a low risk of midges being transported in a trailer or a truck with animals, but unfortunately it's unquantifiable. We can't determine how many midges are actually transported. But good biosecurity is the best method of prevention. So cleaning the trailers regularly, making sure that any muck is removed, any bedding's removed before you start moving animals around, and maybe moving animals at the lowest, at the times of lowest biting risk. So during the day when it's bright sunshine, these midges aren't active and you're less likely to transport them. Other husbandry techniques might be possible, delaying pregnancy until later in the year. As Rudolph was saying, if they tuck later in the year in the north, then there might be a reduced risk of clinical impact of infection. And if a vaccine is available, the vaccine has been used successfully in the past to prevent infection, but obviously it has to be available to be used. And I'll end with a slide. So the Ruminant Health and Welfare Board have made this flyer for Act on BTV3. The measures for Schmallenberg virus, it's the same midges doing the same thing essentially as for Schmallenberg as for BTV. So we can follow the guidance that's been laid out and monitor the Ruminant Health and Welfare and DEFRA pages for any further information. So thank you. I'm going to stop sharing there.
Thank you, Chris. It's an interesting one, isn't it? The comparison between blue tongue and Schmallenberg, and the fact that there are some times of the year when you'd be happy for a midge to bite your sheep with Schmallenberg, maybe sort of when you've not got the rams in and stuff, but we don't want them anywhere near them with blue tongue. So it's thank you for that. Very interesting. I've got a question here from Jan, which was, do midge larvae have a temperature tolerances when overwintering in dung heaps, i.e. in cold winter freezing, or if dung is composted and heats up? I think you kind of answered that question, but
yeah. So there are midge larvae in dung heaps, but they're also in a lot of other places as well. But if we just talk about dung heaps in the winter, dung heaps are actually quite warm because you've got all that decomposing material, and so suggestion is they might be useful sites for overwintering of Culicoides because it's warmer and they can actually be active for longer. But certainly we don't see an impact on midge populations when we have a really cold winter. So it suggests that these insects are protected in some way from really really cold. We know that the frosts knock down the adults, but the larvae are pretty protected where they are. So even when we get very cold temperatures, we still see midges active the following spring.
with that in mind, Rudolph, I mean, with your graph where you showed the spikes in infection and the little one in 2012, if you were to correlate that with the midge infection, I don't know if there's been much testing of midges through that time. Do you think that would have stayed constant, or do you think that's gone up and down as well?
Good question. Because like we say, it sort of maintains itself somewhere through these periods where we don't see it, or we see very few cases. So whether it's a combination of somewhere in the midge population and through the larvae, or whether there's some animals that sort of have it and then transmit it at a low level till we get these explosions. But I think to test the midges itself or the larvae for the presence of virus can be done, but it's quite expensive and quite difficult to do. So it's difficult to do that on an ongoing monitoring basis to actually see a pattern. But Chris will know better how that can be done or can't be done.
Yeah, so Rudolph's right. We can do it, and we can test for Schmallenberg in the midge population. Unfortunately, because there's so many insects and only so much researcher time or funding, that sort of work is really difficult to do. So you can collect thousands and thousands of midges very easily as adults. It's actually really difficult to find them as larvae. in the field. We know they're there because they have to be, but actually getting them getting to them is quite difficult. But as Rudolph saying, it's so expensive to actually test a lot of insects. We might not even get anything back because we don't know if we're looking at the right population at the right time. So yeah, it's one of those things that would take a substantial amount of funding to actually resolve.
Because a constant level of infection in the midges might be a benefit as a sort of natural vaccination in a way to the animals and keeping those peaks from coming up.
Yeah, so if you got so that constant low level infection through the year and not during pregnancy, that would mean that you get immune animals, and that's not what we see with Schmallenberg virus. It does seem to erupt. So where this virus disappears to during the the interim period, as Rudolph was saying, is it just in a few animals? Is it in the midge population? We're just not seeing it. I mean, if it was infecting animals outside of the that pregnancy period, it's likely that we wouldn't see signs, or they would be missed or assumed to be something else. I mean, there's so many viruses or pathogens that, or just all sorts of things that can cause those kind of diarrhea type for a couple of days, milk drop for a couple of days, those kind of fairly generic signs that could well be missed at the other time of year. And it's we wouldn't necessarily see that if there's only a few animals, especially.
I've got a question from Neil here. That do we expect more infection from Yorkshire northwards in sheep flocks this autumn? And we could slightly build on this as well, Rudolph. We were having a little chat before this, and you were saying that you'd had a question from a farmer up north about buying sheep down this way, didn't you?
Yeah. So I guess the concern is if you in an area where you haven't had cases, not on your own farm and not in the area, then you can probably assume that maybe you've got a lot of susceptible animals, but it could also be that they were infected at a stage when they were not going to show infection, and you were lucky in both counts there. But so potentially, yes, because we were caught out before where we thought we had a little peak in 22, thereabouts 23, but then we saw, it started up again and invaded areas where we didn't have it. I think it's maybe a question of timing as well. Maybe by the time, if it gets up going, hopefully it will be too late to affect those animals further north. But there's so many factors that can play a role. It depends how the weather is going to be, if it's going to be enhancing for midges or not. And I think getting back to the question that was put from someone in Scotland wanting to buy sheep from Wales this time of year, was concerned he may buy an animal that could potentially be infected, and then he takes it up to his farm, his midges or his local midges bite that sheep, and then he gets cases from there. But I think I'll leave that over for Chris to answer, because he knows the dynamics of midges better than I can. I do.
Yeah, thanks for that. So yeah, I mean the risk of importing. So we know that the period over which those animals are infectious is quite short. So 2 to 5 days. So if there's some way of holding those animals in accommodation away from midges, if at all possible, especially if it's a small number of animals, that might be more possible. Then you can ensure that those animals, if they were infected, might not be exposed to biting midges. The other thing you could do is actually get serological testing. So if these animals have been exposed to Schmallenberg virus, they should be immune. So if they have antibodies to Schmallenberg virus, they've already been infected and therefore won't pose a risk. Antibodies will come up after the infected period typically. So as long as they have a high antibody titer to Schmallenberg virus, then they should be protected. So 2 things you could do: test first before you move, and if they're negative, maybe keep them somewhere. It's going to be very difficult to know if there was infection from the source farm, because we might not see anything in terms of clinical disease.
Would someone be better off culling the sheep if they've been infected? Or I mean, I guess it depends how bad the lambing's been as well, or scanning, or would it be better to keep them and have a level of immunity?
So if they're still if they're still productive, then they should be immune. So you get sort of long-lasting immunity for Schmallenberg virus. How long it lasts, I don't know, but you get that sort of herd replacement as well factored into that. So by the time we've kind of experienced sort of 5-6 years post an outbreak, we're going to have declined immunity just from new stock as much as any decline in immunity, depending on how long your stock lasts.
Right. Yeah, we've got a couple of questions on vaccines. I mean, it's an interesting one that we jump up and down. We want a vaccine, and then I know with the last in 2011, we all jumped up and down, used it for 1 year, and then it disappeared. And then the poor old vaccine manufacturers. Viv has asked, nice to see you here, Viv. There's no current vaccination for SPV. Is there work in developing the vaccine, and will it come on stream soon? And Jan says, if we have at some stage access to vaccine, what would be the ideal use protocol, bearing in mind the 4+ years cyclical nature of infection?
Yeah, I'll probably start off, like you said, Kevin. We had a vaccine, and it is very effective, but the problem is because the cases then sort of tapered off when the vaccine became available here. So the use of the uptake was quite low, and I think it was also deemed quite costly as well to give the doses as required. And then because these vaccine companies, obviously, it's for them, it's a business decision. So it's a product that took expense to get to market and then didn't sell, and I think that's the problem when it disappears, and when it comes. If it's a very predictable, regular cycle, then you can sort of say, okay, it's going to appear in 24, so start vaccinating the year before. But as we've seen, it's roughly that sort of 4 or 5 year cycle, but sometimes it jumps a year or it comes a bit earlier. So in Australia, where it's a much bigger, it's a massive continent, they do have sort of a prediction program going for these diseases that sort of work for them, but because we're such a small island variable weather, I don't think that predictive model is going to work. So I think if there's a market, then the vaccines can probably be there. I don't think it needs to be developed. I think the principle has been proven. It's just basically making it and putting it on market, but if it's not going to sell, I don't think it's going to work.
Brian would like to know about, we haven't really mentioned dipping OPs, and I'd love to put my Scots hat on and say no, but I have to leave it to the experts, really. Dipping sheep to try and keep midges from biting?
No, yeah, I mean, it's interesting that, we really don't want to be little observations done on farms and by farmers, because it's their stock and they do things and they observe things and they're very good observants of their stock. But I think the story was basically people said they dipped pre-tupping maybe 3-4 weeks before that, and then they didn't get Schmallenberg virus. But considering when an animal would be susceptible to that infection, you're asking that dip to be effective and a repellent for 2 to 3, 4 months, and it's just asking too much of it. So it probably wouldn't have any effect by then. And if you dip too close to tupping, then you're going to get poor fertility just from handling the animals when you shouldn't be doing it. And apart from using it, you've got some side effects of dipping and hitting the environment as well. So scientifically, it's not been proven that is going to have any effect. So it may just be coincidence or just pure luck that it appeared to have worked.
I think so, as a general rule for Schmallenberg, because we see this kind of pattern of kind of neighbours being affected when others are completely disease-free, or counties being heavily impacted and others aren't, all because of that timing of infection. It's really difficult to use the anecdotal observations of people in the field to actually say, well, this will work or this won't work. That's one of the real issues with combating Schmallenberg virus. But as Rudolph was saying, there's no single product or insecticidal use that has been shown scientifically to impact transmission at all.
Like me saying I wore shorts in all of October and I never got it, so it must be wearing shorts that stops it. Another one here. Does midge impact vary if animals are grazing in microclimates, for example, next to rivers or trees? Does this put my animals at higher risk?
So midges kind of tend to cluster around animals, and so where the animals are, there'll be midges. So if you've got grazing near trees and sort of places which are going to be quite sheltered, then you might expect a higher a larger amount of midges that might bite those animals more frequently. That said, all farms vary in the number of midges they have. Some farms, they're super abundant. We haven't found a farm yet where there are no midges, but there's some exposed farms that have very few in their grazing land. So if you have a more exposed field that's sort of slightly open, that would be preferable. Small spaces that are very sheltered would be less so. I mean, they're still going to find your animals. The midges are still going to find your animals. It's just whether you can reduce that by putting them somewhere exposed. Watercourses, such as rivers, ponds, lakes, etc., they're not a habitat for the midges themselves, but obviously the animals come down to drink, and they're likely to churn up the mud and dung next to drinks and cattle drinks and things like that. So although the midges aren't actually in the water itself, they're going to be in the surrounding area, quite happily doing their thing there. But that is true over any farms. If you have a kind of water trough with a slightly leaky tap, that's also a habitat as well. So yeah, so I guess the message is find somewhere on your farm that's the most exposed, and maybe use that land in your rotation as the preferential place to put them in that susceptible period in the first couple of months after tupping or insemination.
Rudolph, why do you think so? A couple of those eastern counties didn't see it at all this year, or you didn't see any case. You didn't actually see any cases.
I think that's the important thing to say, because they had it before. So perhaps if a farm had it again, they said, well, I know what that is, so there's no point submitting it again. I'm sure that played a factor. We've certainly heard that. But it could also be because they had cases that there was more immune animals there. So overall, there may have been less cases than there were in the other more western counties. I think the other concern was as well, and it's anecdotal, I haven't got proof of that, but some people were concerned to bring a deformed lamb to us because they thought if this is blue tongue and it's been confirmed as blue tongue, then they're going to be deemed to be the bad boy that brought the restrictions on their area. And I'm not saying any farmer is guilty of that. It's anecdotal, but we are all human beings, and I can see how people may follow that thought pattern and think, I don't want to be bringing down restrictions on my neighbors.
No, but it doesn't want to be a stigma on it either, really, does it? So people need to sort of keep an eye on things like that. What's just heaven forbid that we get to this stage again next lambing time or whenever, but what is even with blue tongue, what's the procedure for getting postmortems done, if anyone's asking?
Yeah, again, it's obviously if you within a catchment area of one of the APHA submission centres, then contact them and discuss the case with them through your private veterinarian, and the same up in Scotland through SIUC. And then there's also a network of partner post-mortem providers that covers areas which we don't cover. And in some cases, some areas may qualify for the carcass collection service, which is too far away, so then you don't have to bring it. But again, through your private veterinarian, contact one of the surveillance centres to bring it in. I think the thing to maybe put people's mind a little bit at rest, when you see these typical deformed lambs with twisted necks and spinal columns and limbs, as horrible as it is, there's been no reports of BTV3 causing that in Europe. So I think we can become a bit more confident now. If you see that, then it probably isn't blue tongue, it's Schmallenberg. The problem is those that are born maybe like a dummy lamb, or it doesn't have the twisted legs, but it's still affected. And you can see the brain is affected. Unfortunately, blue tongue can do that. And BTV3 can do that. And then the only way is to actually test that. And obviously, if you see anything that makes you suspect blue tongue, that is notifiable. So that has to be notified through the appropriate channels.
Right. Well, our hour is nearly up, really, but that's all really good information. But unfortunately, we don't have any answers, and as you touched on, Rudolph, it is a horrible virus to have to deal with, and the unknowns, and just dealing with it on a mental capacity as well as a physical capacity if you get it. But I'm sure you guys would be available if anyone's got any more questions. They could email you, and I'm sure there's some information on your websites which would be of good detail. But thank you very much for joining us, and thank you everyone at home listening. This has been recorded, so if there's bits you missed or you wanted to go back, I'm sure it'll be up on the NSA website or YouTube channel within a matter of days. They're pretty efficient at that. But again, thank you very much, and thanks Emma, she's in the background there making sure all the tech works. And yeah, thank you very much. Have a good remainder of the evening. Thank you. Bye-bye.
Thank you, everyone.
Thank you, everyone. Thank you. Good night.
Bye-bye.